This afternoon, I'm going to be introducing Matthew Baggott. He will be speaking on the understanding the mechanisms of entactogens and psychedelics in people. Matthew Baggott, PhD, is a data scientist and neuroscientist who has worked to document the effects of psychedelics and novel pharmaceutical agents in humans for over 25 years. He was part of the first team in the US to receive a federal grant to give MDMA to humans, in which he helped develop protocols to safely administer psychedelics and other psychoactives to volunteers. He went on to assist MAPS in obtaining US Food and Drug Administration for the first trial of MDMA in patients. Matthew Baggott went on to conduct the first study of MDA in humans in 25 years and conducted pilot studies with LSD and salvia divinorum A. Please welcome Matthew Baggott.
Thanks. Thank you. Thank everyone. It's an honor to be here speaking at this event, and I hope you'll understand if my talk does not entirely live up to the title. It's pretty much impossible to actually cover that in a good way in 25 minutes. But what we're going to do is something that's hopefully useful and helpful. Before that, a few things to get out of the way. I have no conflicts of interest. Some of the work I'm going to describe was funded by NIH and was supported by a grant to the UCSF hospital. I'm currently at Genentech. However, this represents my views, and Genentech has not seen it, and they would certainly disavow it. So the way I'd like to look at this is think about psychedelic science as trying to take psychedelic experience and make it understandable to the world. So a lot of what psychedelic science is doing is taking this psychedelic experience that's kind of mysterious, and it's taking some sort of established framework that someone else has already made to understand depression or stress or how the brain works, and we're sort of trying to merge the two. And all the scientists are doing this in different ways in their own way. This is super important and probably necessary. One of the things that's good about it is that you can then speak to more people because you don't have to use some language that only trippers know. You can now talk about, like, for example, increased openness, one of the 5-factor personality scale dimensions, and everybody who knows personality psychology knows that. And so that becomes this point of connection between this community and many other communities. But there's risks and dangers in doing this because it's very easy for features to get lost in the translation, and then you sort of overlook them, and that causes the science to potentially have a harder time. And so I want to talk about how we as a field are thinking about the mechanisms of major psychedelics like LSD and psilocybin and entactogens like MDMA, but through this sort of lens of how are we translating it into kind of a conventional framework. And so the 3 frameworks I'm going to talk about are mystical experiences, systems neuroscience and the default mode network, and then entactogens and MDMA as an anxiolytic drug, something that would decrease anxiety and fear. So I figure every good talk has to have an oil painting of somebody that was born 2 centuries ago with some kind of ridiculous beard. So this is John Huygens Jackson, and he developed one of the first theories of visual hallucinations. This was a release theory, and so the metaphor behind this is that you've got stored information in your brain, and it's being suppressed by something. And if that thing breaks, then this stuff pops up. And similarly, if you stop getting sensory input, you also have the same sort of release of perceptions. And this is still a theory that we use today. If you blindfold somebody for a couple days, they will start to see visual hallucinations. Or if people get long-term decreases in visual acuity, it's very common to start seeing things. This theory was first applied to LSD in about 1961 by Jolly West, who was that sketchy guy who killed an elephant and then blamed it on acid. It wasn't really LSD's fault, though. And his rationale for doing it was essentially that someone had been studying the effects of LSD in the thalamus, and they thought that LSD seemed to block sensory input at the thalamus. So like if you took LSD, you wouldn't get any information through your thalamus into the upper parts of your brain, and then maybe stuff would just get released. So that's one theory. A few years later, people in neuropsychopharmacology started talking about serotonin as a filter, with the idea that somehow serotonin is playing a role in preventing you from taking in too much, both externally and also sort of your internal ability to sense things. And that if it gets messed up, what'll happen is that the brain would get overwhelmed. This is a theory that you'll still see today. It's widely used in understanding psychosis, not necessarily just about serotonin. And it got into the psychedelic literature because a guy named Joel Eckes, who is one of the founding fathers of psychopharmacology, was doing EEG research with cats. And he had given them different drugs. And in one case, he gave a cat LSD, and he noticed that the EEG, the brain waves, were much more variable than normal. And he realized that it was because the environment was noisy, whereas with drugs like amphetamine, you wouldn't really get this increased sensitivity to the environment. And so they sort of took it and developed it from there, and it's a theory we still use in many ways. It's a little outdated, though, because now we tend to think about things not so much in terms of a fact that you've stored or a piece of information you take in, but more about we're trying to maybe anticipate things or explore them. And so that kind of change in metaphor can really change how you think about the effects of psychedelics or of how the brain works. And so that's the kind of point I want to try and make in the next 15 minutes or so, that the metaphors that we're using in science, almost sometimes without knowing it, can really condition the way we think about the science and about the drugs. So the first area we can think about would be mystical models of psychedelics, which have been obviously very successful and interesting with the work at Johns Hopkins. If you go back to the early days of psychedelic psychotherapy, there was a lot of focus on essentially trying to create an overall milieu or environment in which you could make a transformative event more likely. So many of the therapists were not thinking of psychedelics as a drug that was a drug effect, but more you created this experience that might be transformative. These pictures, by the way, are from a mid-1960s TV show that filmed Jack Downing, a local Bay Area psychotherapist, giving somebody LSD. And it's online and it's fascinating. So check it out. So in this work, people pretty quickly, or many of them, there's different flavors of psychedelic psychotherapy, of course, but many of them settled on this idea that there was a transformative event that could let people somehow have a breakthrough and an improvement. It went by different names, but the way it's been kind of revived in contemporary research is in this guise of a mystical type experience, a unitive experience, something that's got a sacred quality to it. The credit for that, and I think a lot of credit is due, goes in, to some ways, to these three people. In the center is W.T. Stace, who wrote a book in the early 60s on mysticism, where he argued that there were these consistent features to mystical experience, no matter who you were or where you were. And then on your left, there's Walter Pahnke, famous for the Good Friday experiment, where he actually studied the ability to or the potential of psychedelics to induce a religious-like or a mystical-like experience. And then Bill Richards, who many of you probably know, who's one of our luminaries of psychedelic elders. So Walter Pahnke died tragically young, and Bill Richards, however, carried on the torch and helped Johns Hopkins do the research for which they're well known. And what they found, basically, in their Good Friday experiment 2.0 was that, yes, psilocybin seems to greatly increase the potential for an experience that looks very much like a classical mystical experience. And that was something that wouldn't have surprised Walter Pankey. So here's his description of the first time he took LSD, which is incredibly strong and powerful in terms of what he experienced. The absolute awe, reverence, sacredness. You can read it if you want. So this idea has become justifiably popular and well-known. But one of the things that got a little less attention, I think, as a result of the success of this mystical framing of the transformative experiences, is the fact that even when you carefully prepare people, often people get significant anxiety. Something like 30% with high-dose psilocybin in this group's reports. And this isn't necessarily a bad thing, it just is what it is, but it seems a little sort of incongruous. Last year, the same group came out with this paper where they had surveyed people who had had challenging experiences with psilocybin, and those people, many of them, I think about 80%, felt that they had benefited from the challenging experience. And the researchers looked at correlations and made models, and there was pretty good reason to think that the difficulty of the experience actually predicted or might somehow increase the positivity of the experience in the end, and that the lasting effects were somehow better. I loved this paper because we were finally getting to the point where the field was saying, yes, there's this really interesting stuff that people experience positively, but there's stuff that's difficult, and also here are some ideas on how to navigate it. And that was something that therapists have been talking about forever, but this wasn't something that was really in the hard, cold scientific literature. And so this idea that we should preferentially try to reduce the duration of these challenging experiences, but not really worry about how intense they are. It was a new thing to appear in the contemporary literature. There's some limitations, of course. These are people who had a challenging experience, but then were subsequently willing to fill out a questionnaire, so they're still engaged enough in the topic. So maybe we filtered out some people who had much, much worse experiences. But it's something and it's interesting. And so you could kind of take a simplified model of what's going on like this, and I'm going to draw a couple of these during the talk, where on one side we have these plasma concentrations of the drug versus time, in this case psilocin, which is the active metabolite. And you can see that there's a lot of variability from person to person. The psilocin in your blood gets into your brain and it stimulates 5-HT2A receptors. There's probably a role for 1A receptors and some other things, but let's keep it a little simple, because we do know that if you block this receptor, you really don't get the experience. So the other ones are probably modifying the experience, but this one is necessary. And then if you're lucky, you get a mystical type experience. If you believe in set and setting, you might try and throw that in as well. I sort of don't. I tend to think that something like bedside manner is what's really important, and set and setting too often emphasizes the sort of initial stuff, not the interactions, which I think are core. But you sort of can have this simple little model. You have the experience, and then you have this lasting positive effects. You say that was meaningful, and you feel more open to new experiences. And now we can kind of modify that a little potentially. We're saying, okay, sometimes there's a difficult experience, and there's another route you can take to basically the same effect. And there might be maybe some lasting meaning that you get that's not because of the mystical experience, but just because you overcame some kind of challenge. So the question I would have is, were we sort of maybe led astray or slowed down by calling it a mystical experience? In one sense, obviously, if you have a mystical experience questionnaire and the person fills it out and they get a high score, by some definitions, it is that. And so it's more a question of, is this yes and also other stuff, where we were kind of overlooking the richness of the phenomenology because we had a specific model we were using. So that's psilocybin and mystical experiences. The other thing I wanted to talk about a little was systems neuroscience and the work that's being done mostly at Imperial College on this default mode network theory of psychedelics. So serotonin is something that has a lot of control over slow cortical rhythms. So like your neurons in your brain, in your cortex, they can get closer and further from firing depending on how much input they're getting in, and they sometimes have their own little preferences, and so they kind of oscillate around, getting closer or further from firing depending on whether there's pumps turned on and off. This kind of stuff turns out to be really useful because we think that the way the brain connects one part to another is through correlated oscillations. If you think about it, there's no real switches, so you can't be like, all right, turn that off, turn this on. Instead, you've got to be like, all right, we're all synchronized together so we can talk, and then you're at a phase, so I'm going to ignore you. And so something like that happens. This is a fairly abstract demonstration of that, where what's being plotted is LFPs, or local field potentials, which is just kind of the summing of lots of inputs that lots of neurons are getting. It's being measured with an electrode. And the neurons seem to go either into this up state, where they're close to firing, or a down state. And they're going kind of back and forth between the two of them over time, which is on our horizontal axis. At some point in part 2, the researcher has stimulated the cell bodies of the serotonin cells in the dorsal raphe nucleus, the DRN. And what happens is you get this profound change in behavior up in the cortex. And so this is sort of a simple demonstration of the way that serotonin can influence the behavior of the cortex overall. Serotonin in general does what we call volume transmission, where it gets released, but it doesn't just signal the one synapse, the one receptor, the one neuron right there. It kind of flows out and bathes the cortex. And so it's in a very good position to kind of modulate the overall excitability or sensitivity of the brain. If you want to talk about important brain parts and unimportant brain parts, they're all pretty important, but you could sort of decide to pick this area here as a reasonable starting point, the precuneus and the area around it. Seems like it's somehow the center of a hub that's important for maintaining awareness and consciousness. And so if you look at the difference between when someone's conscious or not conscious, there's a pretty big difference. This area turns out to be part of a network that we've all been hearing about and studying. So if you measure brain activity in any way, really, if you just get enough data, you can just ask yourself, which parts tend to be correlated in activity? If this is active, is that part active too? And if you do that, with certain assumptions, you can kind of segment the brain into these networks. And so we've got that on your right, these different networks you can kind of automatically find. One of them is a default mode network, which is shown in more detail on the other side of the slide. And this seems like it's active when we're kind of doing nothing, thinking about stuff, just being ourselves, remembering yesterday, maybe thinking about tomorrow. It's a little mysterious, but it's the thing that if you don't ask someone to do anything, this tends to get more active, and if you ask them to do stuff, it often gets less active. Now, these networks are real in some senses, but we should keep in mind that they exist because we've decided to break the brain data in a certain way. And so there's assumptions in how we decompose the brain into networks that cause us to consistently get these, but at the same time, it's sort of like maybe boundaries for states and nations. Like, yes, it's true, but it's also, we could have done it a little differently, and it would be a little different. So, I don't know why I'm pointing at the monitor. It's probably over there to point. So a really exciting finding over the last 2 years in a couple different papers has been that LSD and presumably other psychedelics alter functional connectivity, and that these connectivity changes correlate with self-report measures, including ego dissolution. So that's really cool. And we can go from that and to start taking measures of connectivity. And so we can say, all right, let's look at the default network and just look at the correlation of all the little measurements we made within that part we've defined as a network and see if they're more or less correlated on a drug versus not on a drug. And we can get an effect. It can be significant, and we could call it, if we want, disintegration. In a way, what we're doing now is we're taking our theories about psychedelics, and now we're sort of imposing them on systems neuroscience and how neuroscience thinks about things. And it's fine in some ways. There's a potential danger because obviously not every decrease in default network connectivity or default network correlations is really a disintegration of the ego. But maybe it is in this case. I think it's an open question. And so we might think about this model as being something like, you give LSD, it's in your blood, it goes into your brain, stimulates these receptors, and again, we have a pretty good idea it's these receptors because we've blocked it and blocked many of the interesting LSD effects. And so it seems like it's needed, but there might be other things involved. And so the theory is then the connectivity gets altered and the ego dissolves. And also, if I'm reading this theory right, you get some sort of residual changing in your brain network functioning that has positive effects, maybe increases your openness to experience other things. It's a good theory, but one of the hardest things for us to do in these kinds of studies is figure out the chain, the sequence of this one goes before that one. For most of what we do, we're kind of like squinting and doing correlations and stuff like that, and it's possible we're wrong. It could be something like this, where really the connectivity stuff we're measuring has nothing to do with the beneficial effects. Maybe it's just the phenomenology, and our brain measures are not measuring the right thing. I'd be hard-pressed to make a bet on that one. And if I were forced to flesh out the idea, it would be something like, well, yes, I think that there is a change in connectivity associated with ego dissolution. I just don't know that we're measuring it with this sort of measure, partly because when A receptors are going on, and they do stuff and other things. And so we could kind of step back, because I don't feel like this research group is doing a bad job or anything. I think they're doing as well as anyone in the field. And think about it this way. Drug concentrations predict the intensity of effects. So here's a study on one side from George Agajanian in the 60s, and here's a recent study from Patrick Dolder and Yasmin Schmidt on the other side. And in both cases, you can see that there's a pretty good correspondence. If you measure the right effect, they're tightly coupled. One problem is that LSD concentrations vary a lot. And so you've got like a twofold difference between people who've been given the same dose orally. The end result of that is that if you try to do a correlation, you're going to end up with a possibility that since each dot is a person, you're just really measuring some people got small doses and some people got big doses. And the way you try to protect yourself from this is you do a bunch of correlations, but that's not going to help if the measures you're using are different in how sensitive they are or how noisy they are. And so we end up in this realm where we're interpreting something like noise. And so I think as we move forward, it's going to be important to try and deal with that. So I'm going to skip quickly through MDMA. And basically, the story was, it sounded like an anxiolytic, but it increased anxiety in the lab. And it also made people less relaxed. We found that it lowered social anxiety and made you feel okay being yourself. And so you can take a bunch of Arrowwood reports and say, okay, let's analyze them and find topics automatically. And one topic turns out to be like love and beautiful, and one is like really good. And if you plot all the drugs, you end up seeing entactogens in a certain spot, or things that are sold as entactogens. This seems to fit the idea that these drugs that have much more serotonin release than dopamine are kind of in the MDMA camp, as long as they're also releasing norepinephrine to make them a stimulant. And so we've got this kind of stimulant row here. So if we think about this, we could also try and put it into this sort of framework, and you end up with something complicated like this under the old system. But I would say that really what we have is something like this, where MDMA is releasing norepinephrine and serotonin. That makes it both sort of positive mood-like, but also changing how we process emotions, causes oxytocin to release, and it does stuff. But what's important is that it doesn't actually impair emotion, people are still able to be appropriately emotional on MDMA in my experience. Reports that we've seen in the lab that there is some sort of like worsening of tests, like if you have people recognize emotions, I haven't yet to see a single test like that that corrected for guessing and really did a good job of showing that it wasn't just some non-specific impairment or mood congruence. If you feel good, you don't guess negative emotions. So that's kind of where we are with this. And overall, I've tried to say that framing psychedelics in these ways can be helpful, but also risky. And one of the best things about doing this is you start to get new ideas. You're like, oh, I never thought of a psychedelic in this way, and I have a hypothesis. And one thing that jumps out at me when I look at this all together is that the mystical experience seems to predict you need certain experience. The default mode network seems to really not make a prediction about that, as if you could give someone LSD in their sleep and have a beneficial effect. So that gives you testable things like that. Not that I'm advocating that, but I mean, if you got IRB approval, why not? And so overall, I think that we benefit from thinking carefully about what are almost metaphors that we're using, that they just get into our skin and they don't sound like metaphors anymore, but they really do guide and shape the way we think, and so let's use them productively. Thank you. Mike? Yeah, so those slides were made late last night, and I'm happy to make them available. I think they're streaming though, right? I think the video is being
everyone should have the
streamed.
opportunity to see where Sasha's lab was, because
But-
we don't know how long.
but I'm happy to
that's
email
gonna
them to anyone
be
as well.
in her alive. and
I was just wondering if you
willing
could
to
expand a
have
bit on
these
the slide
gatherings.
about the MDMA and the emotion
Thank
recognition.
you for what you
Because
do.
you kind of skipped through it, and I was wondering what...
We're all in it
you were-
together, right?
Sorry, I was wondering if you could expand a bit on the MDMA and the emotional recognition, and...
Sure.
Yeah.
So if you do an emotion recognition task, you typically use the standard Ekman emotions, and the emotions vary in terms of how distributed the information is on the space. And so fear is a little harder than some other ones because more of the information is in the eyes. If somebody's spatial attention is impaired, they might look in the wrong place and miss it if it's a brief stimulus, things like that. So
Yay, she
you
made
would
it.
ideally control for that kind of thing by having some other task with focused spatial information versus diffuse spatial information. Secondarily, what you would want to do is you would want to say, how often did they, out of all their guesses for fear, how often was it actually fear? And do these sorts of corrections. And so D prime is the standard signal detection measure. And so what people typically do when they report is they just give the percent accurate rather than correcting for the pattern of guessing.
I'm so sorry, it's on this side across the street from Foz Restaurant. Foz is one side.
Thank you, everyone.
Hotel on the other.